Hello, we are together with Zoe Cournia. Hello, Zoe.
Hi, nice to meet you.
Nice to meet you too. Welcome to Cluj. You are running a group that has developed a machine learning web server, right? DREAMM. And also AlloPockets, another deep learning model train. Before going into those details, tell us about congratulations. First of all, congratulations for winning the prize this year as ISQB, and tell us a few things about the paper you present today. I mean, here.
First of all, it's a real honor to be awarded with a Gilda Lowe lectureship. She was an American chemist, a pioneer of computational chemistry back in the 90s. So it's a really big award, also being a woman in computational chemistry. That's really important. So in our lab, we work to connect the molecules, the proteins, with their function, with the ultimate goal to produce new candidate drugs. For that, we use machine learning, and we also use other techniques of physics-based. And the software that we produce is basically to help other scientists as well to incorporate these methods in their workflows. So what we are presenting today is machine learning models to aid the elucidation of pharmacological targets and how these are connected in the cells and how we can produce new knowledge to identify where exactly we should place drug molecules on the surface of proteins so that we can have later candidate drugs that can go into mice preclinical models and maybe later also into humans in clinical trials.
Sounds good. So this, if we'll have another pandemic moment, this should help us, right?
It should definitely help us, and one of the major aspects is open science. The codes that we write and the software that we develop is all open source and open access so that everybody can use it. And as you mentioned, during the COVID pandemic, the vaccine was produced so fast because the whole worldwide community came together, provided free access to the data, to the structures of COVID, to the drugs that can be used for COVID, and all of this brought the knowledge of the vaccine so fast. So we are real believers of open data and open source software.
Sounds great. And tell us more about your DREAMM machine learning. How many? There is one model, or there are many models for?
Yes, actually, there are many models that we can use. One model we produce when we have data for the structure of the pharmacological targets. Then we can use the structure to predict how this protein will bind on the cell membrane and how then we can identify pockets to produce candidate drugs. But we also have other versions that use large language models. When we don't have structure, we only have a sequence of the protein. These large language models can actually predict this information where the protein will bind on the membrane without having the structure. So there are different versions according to what data you have available.
And one more question: how do you see the future of medicine for, I don't know, in 5 years or maybe 10?
So the future of medicine in
And
conjunction
drugs, yes.
and drugs in conjunction also with computational chemistry, I am not so optimistic as the the Meta-AI or the Bold people. Google always advocates that in 5 years we will have solved all diseases
and
we
live
will have
forever.
yes, we will live forever, we will have all drugs. I don't think that this will be possible because there are intricate details that are not yet encoded in machine learning models, and it will definitely take more physics-based models to uncover all the molecular details of life. So I am optimistic that computational drug design is helping our advancement of drugs, but we will not solve everything in 5 years. And we'd also like to thank Vlad Koziokaru, who is the current president of ISQBP, for giving me the opportunity to be here and awarding me the lectureship. And also Modesto Orozco is a pioneer in our field and has been awarded the Computational Biology Award. And the new president for the next term, Marco De Vivo, who is also here and supporting these activities.
Thank you, Zoe, for coming inclusion. I hope you will come,
Yes.
more often than this. You will share with us your
Yes.
Research.
Thank you for inviting me here, and congratulations for the event and covering also for the magazine. And I hope to see you again.
Thank you.